We'd like to remind everyone that this webinar is being recorded and it'll be made available on the Opentrons website. We'll also be sending a copy of this webinar presentation and recording by email. To help with the overall webinar experience, all attendees are on mute, but to ask a question, please use the Q&A box on your screen, and Curtis and Kennery will answer your questions at the end of the talk. Today we have two great speakers, Curtis Knox from seqWell and our own Kennery Watson. Curtis is seqWell's Director of Product Management and brings more than 19 years of experience in developing, marketing, and selling genomics reagents, including products for NGS amplification and synthetic biology applications. Kennery is Director of Science at Opentrons, where her team works cross-functionally to develop robots, modules, and software for lab automation and verify automation protocols for assays across genomics, protein-based, and cell-based applications. We're very excited to hear about ultra-fast, high-throughput multiplexed library preparation of plasmids and amplicons using seqWell's ExpressPlex chemistry on the Opentrons Flex. So Curtis, over to you.
Thanks so much, Andrew. I'd like to start off quickly by going through a short agenda of what myself and Kennery will be discussing today. First off, we'll start off with a brief description and some reasons behind why the need for high throughput plasmid sequencing is becoming so important. And we'll top that with an understanding of the ExpressPlex chemistry and why we released that for high throughput library prep. And then I'll turn it over to Kennery, and she'll be able to describe the Opentrons Flex platform and how we actually automated the ExpressPlex chemistry on the Opentrons Flex, including some results and some data and understanding about what we were able to actually achieve. And then we'll finish up with some descriptions of what our future looks like, and then open it up for questions at the end. So first, I want to talk a little bit about the synthetic biology discovery process and everything that goes along with that. From a perspective of thinking about the process overall, it really becomes a kind of a rinse and repeat situation where you are constructing synthetic sequences of interest, be it in plasmids, amplicons, however you want to do it, and everything that goes along with doing that. And you want to be able to sequence that quickly and analyze that information, and then just continue to turn that around as much as possible, such that you are looking at your constructs and doing a lot of those as quickly as you can and as many as you can, so that you can get to the data at the end. And in needing to do that, you can need to do thousands of those constructs with every round that you're looking for. And with that in mind, we designed the ExpressPlex chemistry, which is a situation or a kit where we really thought about the high throughput needs of these types of customers. And with ExpressPlex, we've designed a method by which 90 minutes start to finish, and with only about 30 minutes hands-on time if you're doing this manually, in which the fragmentation of the DNA, the barcoding of the DNA, and the amplification of your library happens all in one step. And with that, you have primers, and your barcodes and everything are built into the product. You don't have to go out and buy those separately. And it allows us to actually be something where we're getting up to 1536 barcodes all ready to go, and you can do all of that, prep those libraries, and actually sequence them in about 24 hours from start to finish, as far as that goes. And the workflow, as it looks like with this product, is as follows. You've got an indexing plate that's provided in the kit, and that contains all the barcodes, everything. You've got a ready reaction plate, which is the master mix, and then you've got magnetic beads for the cleanup. All you have to do is supply your DNA plate, and the workflow is this. You transfer 4 microliters of your DNA into the ready reaction mix. You transfer 4 microliters of the indexing reagent into there, and then you mix those, put them on the thermocycler for an hour, and at the end of that, you are simply pooling all of those samples into one tube and doing a single cleanup on a single tube to then move forward to your sequencing. Now, what does this look like? And what are some of the things that we can achieve? And one of the great things about this system is just the sheer, small number of reads that are required for you to actually get your plasmid de novo assemblies. And we have shown that even at 500 average read pairs per plasmid, we're getting 100% error-free circularized assemblies of these plasmids, compared to at those low levels of reads with Nextera XT, either in full strength or at a quarter reaction, you're still losing a fair number of the assemblies. And what this translates to is minimal flow space required. So you can actually continue to multiplex and put in more and more samples onto a flow cell to bring down your overall sequencing costs at a much greater level. And we're still achieving really nice success rates for the plasmid assemblies. Along with that, one of the other really great features of this product is an auto normalization feature of ExpressPlex. And I want you to think about, how variable are your samples? Do you have, a wide range and you're trying to do individual normalization of those samples such that you're spending a lot of time and a lot of plastics getting ready after your extraction to the point of actually being ready for the library prep? With our system, it easily tolerates a 5 to 10 X range or fold input. And simply, if you've got a process where you're getting samples in a given range, you might just need to do a simple global dilution across your plate. And you can just test a couple of them, or if you've got a system where roughly where everything is coming out, just simply do a global dilution, get them in the right range, and then that allows you to have an even faster overall turnaround time and using fewer plastics as you go. So here's an example of a situation where ExpressPlex, across a wide variety, in a 10-fold range here, you're seeing very equal read depths across these, whereas a similar input range, at least from a fold perspective, for the Nextera kits, you see a much broader range of overall outputs of read depths that go along with it. So now I want to take a moment and turn this over to Kinnary and let her talk about the system overall and how that works for there.
Thanks, Curtis. So for a bit of introduction, the Opentrons Flex is a platform that we launched last year and really has all of the components required to automate chemistries like ExpressPlex in a really robust fashion. So there's a lot of modularity and flexibility in the pipetting heads that can be attached to this liquid handling platform, as well as the features, grippers for transporting labware across modules and to trash and elsewhere on the deck. There's also an on-instrument display, so you can really easily set up and run your protocols with very minimal changes per run. Additionally, we do offer bundled solutions, and I'll talk a bit more about those features in a few slides that can help with you deciding on the components that you need for your particular NGS application. So we're really optimizing for walkaway time and making this a really seamless experience for automation. To be more specific about some of our features on the next slide, we can just zoom in on the pipetting capacity and efficiency that we can present on our platform. So we have a single and 8-channel pipette. You can have 2 pipettes attached to the platform per protocol, and those volume ranges are 1 to 50 or 5 to 1,000. So really broad ranges of supported volumes for each. Also, the 96 channel pipette can be attached in lieu of these two pipettes, so 5 to 1,000 microliters, which is another kind of broad range that has a lot of flexibility for various applications. So that can be modulated dependent on your preferred efficiency or protocol complexity there. And so here we're just showing a bit of the specifications to really illustrate how compelling and competitive the efficiency of liquid handling can be on the Opentrons Flex. So on the next slide, we can really zoom in on the components that we're going to be talking about that actually we utilized for this particular chemistry, the ExpressPlex chemistry. And what we see is that we used a P50 multi-channel pipette as well as a single P1000 pipette for pooling the samples together. And he really laid out the the workflow, and we'll show a bit more about that on the next slide. But the workstation itself also has a lot of the components and labware that you'll need to get started. We also can include custom protocol development for NGS applications. The great thing about this partnership with seqWell is that we've done the heavy lifting of the protocol development ourselves in all formats that could be possible for Flex automation. So as Curtis mentioned, there's a really simple workflow that makes it really ExpressPlex really amenable to automation. And so with the components I mentioned, we configured a deck design that enabled the pooling plate, the tip boxes of those varying volumes, as well as the amplified reaction plates for us to really simply prep the plates and then pool the samples for the subsequent sequencing steps. So on the next slide, is where we actually start to get into the meat of the results here. So we were really planning to recapitulate what ExpressPlex has already been shown to do, which is really construct high yielding libraries uniformly. And so we used PUC19 with the configuration I mentioned, and we compared against manual results at the recovery or yield of the libraries that we constructed on the Opentrons Flex. And here you'll see that we were able to meet and exceed that particular experiment. So we're able to get really high yielding libraries constructed on the Opentrons Flex. Here, this is just PUC9. So we're getting what we want in terms of the yield and recovery out of the samples that we prepped on this platform. So as we go to the actual sequencing results, which we're all compelled to take a look at, here we're also, for reference, we were sequencing those plasmids. This is per 96 sample set. And this is of PUC19 on our NextSeq 2000 with 2x150 reads, paired reads here. So for those that are passing the read filter, we're exceeding 98% here on average. So we're getting really high quality reads. And so we're getting the most informative capability we can out of those samples. And then in addition to that, we're getting a higher Q30 value. So most of the proportion of samples, specifically around above 92% in terms of the actual Q30 threshold there. And then we have another snapshot on the next slide about the balance of samples. This is another way that we just recapitulated the really strong performance of the seqWell chemistry. So across a plate here, we're showing a few seqWells, in addition to the controls. But we're able to see that it's really balanced across the sample plate. So that 11.5% is really as low as we can illustrate here. So we're really comparable to what the chemistry can show manually and what the power of the actual kit here. So with that, I think we also want to discuss the efficiency. So this particular workflow, as I was showing with the deck layouts earlier, is really condensed. So we can really optimize for a really quick turnaround in terms of the time from sample preparation to actually getting your sequencing results off your sequencer. So we can prep for reaction plates, and we can actually do that sequentially to meet the need of, let's say, up to 768 samples, being a great example of that optimization. And so with sequentially actually starting that experiment, because it can be done so quickly, we can stagger that and four amplify four plates, and also pool those plates and get those cleaned up in time to get them on the sequencer to have a turnaround time of around 24 hours or less, potentially, depending on your particular sequencing need. So taken together, we can really optimize this particular solution with the automation and with the robust, condensed, powerful chemistry of the ExpressPlex kit, and really get samples sequence to sample really quickly. So I think I'll just highlight here a couple of those takeaways from the automation aspect of this project. Those PUC19 samples are really illustrating high uniformity. We're able to have the samples be really uniform across the plate, and then the subsequent samples, the sequencing reads are really high quality and informative, mapping back to the samples really in a high fidelity fashion. So we're able to really have some high quality of data produced on the automated workflow. In addition to that, we can optimize that even further. So we have a couple of plates that we can process in sequence to really get from your plasmid to your sequencing results extremely quickly, and really competitively in the landscape of prep of plasmids. And then, of course, there's quite a bit of optimization that we can do for different formats. These were 96 formats, and I'd love to hand it to Curtis to talk a bit more about even higher throughput capacity.
Yeah, thanks, Gary. And that teed me up pretty nicely here. So, as we continue to talk to our SynBio partners out there, as ExpressPlex and the automation through Opentrons and everything else has come out, it's been very obvious that the users of this type of platform want to go even faster. They want to go even bigger with what they're doing. And with that in mind, we already have it in early access and will be fully releasing as an off-the-shelf product in March, what we're referring to as ExpressPlex HT. And this is actually the first ever commercially available 384 well library prep kit that's actually enabling, and we will have 6144 barcodes available for this. And we do have users now actively doing 6,144 samples at a time. And really what, again, what happens in this is if you think about trying to multiplex to that level, you would have to go out and source all of those barcodes, all those primers, and put all this together into a 384 well format. That takes a lot of time and effort for someone to do that from scratch. Instead, we're providing a kit that allows you to do that ready-made, and really thinking through similarly what we had in our workflow before is something that's now actually even easier in that we've redesigned the workflow of this, and so that you are pipetting on the left-hand side there, you see from your 384 well plate of DNA, or you could start with 96 well plates of DNA, and you're pipetting that into the indexing plate where everything is already pre-arrayed in 384 well plates. You're pipetting from the DNA into the indexing plate, tip mixing that, and then using that same set of tips and transferring another 4 microliters of that directly into the ready reaction mix plate. Again, sealing that, thermocycling, and then pooling all together in order to do 384 samples at a time. And we've built out 16 different barcoding plates so that you can do 6144 in a staggered situation. And the really amazing part of this, and this goes on top of what we talked about earlier, in that combining 6,144 samples at a time on a MiSeq or a NextSeq, and we've done both, you can actually bring your physical sequencing costs per plasmid down sub $1, easily as little as 20 cents a sample for your physical sequencing costs. And we are actively working with Opentrons to, this will be the next step in which, I don't have a timeline yet, but we're working to start, start building out this workflow for everyone as well. And in the end, with this, really what we think about is accelerating what many people refer to as the design, build, learn cycle. Think of this as an entire workflow for high throughput plasmid and synthetic biology discovery, in which you're starting on the left-hand side into the design, where many laboratories are using machine learning or AI to design new potential constructs. They're building those constructs, they're transforming them into plasmids and creating and growing those up. And then they need to go on to sample prep and everything else. So really where we're continuing to accelerate this is now, extracting that many samples at a time is very difficult. What we have seen is that by using colony picking, you can go into colony PCR, or you can use rolling circle amplification in a way that you can just build up the DNA really quickly. So you can either sequence just the insert, or you can sequence the entire plasmid. It really doesn't matter when you're going directly into ExpressPlex from there. So you can greatly accelerate this entire cycle of learning, such that when you've got your colonies and getting to the point of actually having sequenced DNA off of all those colonies, you can still achieve that in roughly, a next day turnaround time. So this is extremely exciting when we think about just how much we can accelerate the overall discovery process for our high throughput customers. And this can be for antibody discovery, this can be for site-directed mutagenesis platforms. There's so many different applications where something like this can be very, very useful. So with that, we want to pause. We're going to have a Q&A session here at the end. From my side, I would certainly like to acknowledge, our team, John Palis, who is our automation specialist at seqWell, and then Becca, Stella, Jenna, and Jack, who's our co-founder and CTO, who are all the inventors and very strong R&D team who has been behind the genesis of ExpressPlex. I'll turn it over to Kinnari for her team, and then I think we've got one more slide, and then we'll open it up for questions.
Right. Yeah, I think the most important is Deandra Howell from the team, who really scripted out and did all the analysis for the sequencing data that I presented here today. And then, of course, our counterparts, Brent Barton and Fran Slater from the rest of the Opentrons team. So I think with that, we'd love to invite you, if you're attending SLAS, to come and stop by at Opentrons booth 1216 in Boston. If you would like to scan that QR code, you can arrange a meeting with some of our team members. And of course, you can reach out to us on our website as well. And for you, Curtis?
Yeah, so we're not exhibiting at SLS, but we will be running around. I will personally be there Monday through Wednesday, as well as several of our sales team members. So if you want to reach out to us via email, just contact us at sales@seqwell.com, and we will be happy to arrange a meeting for you out there. Anyone who happens to be attending AGBT next week in Florida, we will be there as well. So we'd love to meet up with you, or if we can't meet you there in person, we'll be happy to set up a chat for you right away from that perspective. So we look forward to either meeting you in person or talking to you about not only Opentrons Flex, but ExpressPlex, and how we can put all these two together so that we can continue to accelerate everybody's synthetic biology discovery. So thanks, and looks like we've got a question or two here. The first one I see, and I think this is more of a question for you, Kinnari, is can you pool the samples into one tube on the Opentrons Flex platform?
Yes. Yeah. So we used our higher volume single channel pipette for the pooling steps here. So yes, it's really feasible for us to do on the Flex for this particular experiment. We used that P1000 single channel pipette.
Mhm. And next question, is ExpressPlex HT currently available? And the answer is yes, it's available in early access now. So we've got 1536 of this we could put in your hands right now. And it can actually do a little bit more than that. The full 6144 group of products will be launched in mid-March. So the answer is yes, we can absolutely help you there. Looking through additional questions here. Do you think your chemistry, primers and master mix, will work for generating whole genome library for NGS? The answer is no, not yet, because it is a little bit lower complexity product. It is designed for plasmids and other synthetic constructs. It does not currently generate a high enough complexity library for large genome, whole genome type applications. There are future discoveries and advances to come from that perspective. Next question from Jessica. What is included in the preloaded reaction plate? How many barcodes are available for the current 96 plates? And then there's a follow-up question for you, Kinnary. So in the preloaded reaction mix, that is basically a master mix that allows for all of the amplification piece of it. The tagging reagents include preloaded TN5 transposase with the different barcoded reagents to it. And the current 96-well version has 1536 barcodes available. So there are 4 different versions of that kit with 384 barcodes each, and those can be multiplexed together for 1536 barcodes now. And then there was a question for you, Kinnary, in that same one. There are the Opentrons Flex pipettes compatible with the Opentron 2?
Right. So the short answer is no. If you have an OT2, you would have to use those OT2 compatible pipettes. So you can reach out to us if you need some additional resources for scripting this for an OT2 or any kinds of additional adaptations to make, but we would have to change the format a bit.
Sure. Okay. Next question from one of our attendees is, does seqWell offer data analysis post-sequencing? We do not yet, although we do have a pipeline that we have designed specifically for plasmid assemblies. And that is something that we're looking to make more available to our customers through a variety of opportunities. So we're happy to sit down and talk to you about how that could potentially work. So I think that's something we can help you with. Question from Chris Boyles. What is the typical plasmid size that the kit is designed for? We have worked with a variety of plasmid sizes. He asked 4 kb, 10 kb, larger. We have definitely worked up to 10 kb with no issue. I'm feeling confident we can go above that. The way the overall chemistry works, larger is actually a little bit better here. So again, feel free to reach out to us with your particular plasmid size, and we can walk through with that. Question is 20 cents per sample the cost per library prep or sequencing? That is referring to the sequencing cost. So if you divide a cost of MiSeq or NextSeq flow cell, which is generally in the $1,000-ish range for the capacity required to do that many samples, and divide that by 6144, that's where I'm getting that number. That does not include the library prep cost. Does seqWell have a PCR-free WGS kit for human mouse samples? We do not at this time. Is ExpressPlex Plasmid compatible with ONT sequencers? The kit that we've described today is around Illumina platforms. We do, we are exploring other opportunities in that space. So again, would recommend that you reach out to us and we can have some conversations with you around some of that. We are not beholden to a particular sequencer. Obviously, Illumina is the dominant one, but we are certainly looking at how we can potentially improve and expand the overall opportunities there. So I think that is all the questions that I have on the list, unless I have missed one. Andrew, do you have any more that need to come through for Kinnary or myself?
I see one more that came through, that you talked a little bit about the throughput possible with this system, which was very impressive. How does that compare with a typical throughput doing these manually?
So, I mean, you can do the 96-well one, you can absolutely do manually. It will take you longer to do that, and somebody might get carpal tunnel there. It's the reason, of course, for using an Opentrons Flex, and automation in general for there. So, for that one, you can do in either fashion. And the throughput will obviously be a little bit quicker, a little bit more efficiently using an automated system. For the 384-well ExpressPlex HT, we would never recommend trying to do that by hand. That is a recipe for disaster, regardless of what chemistry you're trying to work with there. So, a situation like that absolutely requires an automation platform.
Well, I think that wraps up all of our questions. So thank you very much, Curtis and Kinnary, for such a fantastic webinar. And a quick reminder to everybody that we will be posting the webinar on the Opentrons website and sending it out via email. So thank you all very much for attending.